A very good morning to you all. Welcome to our first session of class 11 bio, and today we shall discuss that is very familiar to you all, that is cell. Now, as you know, cell is a structural and functional unit of life. What you have studied earlier also, but there's a slight difference. You will be asked like of the contribution of the various scientists, 1 marks questions. So you have to remember certain contributions of various scientists. Some of them are like Robert Hooke, who discovered the cell. Then you have to know about Antony van Leeuwenhoek, who proposed that the cells are living. What Robert Hooke had discovered, the cells were dead, since he had observed the cells of the cork cells which were dead. Then we have Robert Brown, who discovered nucleus. Then you have another contribution, that became the fundamental aspect of the cell, that it is a structural and functional unit of life, that is Matthias Seliden and Theodor Swannen, both German botanists and zoologists, who proposed the cell theory in 1839, and that was modified later on by Rudolf Virchow, who proposed that all cells exist from pre-existing cells. Then we have another contribution of J. Prokanji, who coined the term protoplasm. Now, we have already discussed what is cell. Cells, you all have studied earlier also, that they show variations in shape, size and number. Now, why do they do so? Because of the functions that they perform. Their shape may be cuboidal, spherical, polygonal, and their number also differs. The size also differs. The smallest cell is mycoplasma or PPLO. Full form is Polyribosomes, that is like organisms, which are around about 0.3 μm. Now, what is μm? It is 1,000 of a millimeter. Then you have the bacteria cell, that is also considered as, the size is considered as very small, around 3 μm. You have the RBC cells, about 7 μm. Then you have the nerve cells, which are very long, about 1 meter long. And then you have, of course, the largest cell, that is the ostrich egg cell, that is about 170 into 135 micrometer. So these all comes in 1 marks questions. Clear? Now, when we talk about the cell, there are two types of cells, prokaryotic cell and eukaryotic cell. Today we shall discuss about prokaryotic cell. Prokaryotic means what? Pro means primitive. A cell which has a primitive nucleus is called as prokaryotic cell. Cells, that is, these are usually found in the members of kingdom Monera. Monerans means what? That is the members of the bacteria. That includes the archaebacteria as well as the eubacteria. That will be studying later on. That is, and then you have cyanobacteria, also referred as blue-green algae. Now, what is the important features of a prokaryotic cell? Most important thing is that, as I said, nucleus is primitive. Primitive means the nucleus is not well defined. It is called as nucleoid. You can see here in the diagram here. Come closer. So you can observe that the nucleus is not well defined and it doesn't have nuclear membrane, nucleoplasm or nucleus. That's why it is referred as nucleoid. Sometimes an extra chromosomal strand of DNA is present, which is called as plasmid. Clear? Now, when we talk about the prokaryotic cell, like the bacterial cell, what features do they have? First of all is the cell envelope. Clear? Now, we have studied about plasma membrane or cell membrane. But here, in the prokaryotic cell, what do you find? That is the cell envelope, which is the outermost part and consists of 3 layers. Clear? The first layer is the glycocalyx or mucilaginous sheath. Sometimes the bacterial cell, the outermost layer, is slimy or having mucilaginous sheath or a very hard covering called as capsule. This is to avoid, or that is tied over harsh conditions and also protective in nature. Clear? Now, next comes the cell wall. What is the cell wall made up of? Cell wall is made up of peptidoglycan, or also referred as murin. Clear? That also it allows the entry and exit of substances. Then it also provides protection in bacteria. Now, depending upon the according to Christian Graham, the cell wall can be stained according on the basis of certain dyes. They are grouped into 2 types: gram positive bacteria and gram negative bacteria. What is the technique of gram staining? Actually, what happens, that is, when the bacterial cell is stained with crystal violet dye, it stains blue. Then it is treated with iodine and then with alcohol. So if when observed under the microscope, if the cell wall retains the blue color, it is referred as gram positive bacteria, and if it does not retain the blue color, then it is referred as gram negative bacteria. So again, like I said, like while I am teaching you all, I'll be also emphasizing on the topics which can come as objectives, which will be asked in detail. So sometimes you may be asked, like, what is the difference between gram positive and gram negative bacteria? Gram positive bacteria, what is their characteristic features? That is, since they can be stained with gram stain and they retain the blue color, they are referred as gram positive. Other features are what? That is, they have around 80% of cell wall is made up of peptidoglycan, but the outer membrane is absent, and of course lipopolysaccharides are absent. And one more characteristic features is that is they are sensitive to antibiotics. Clear? Like cyclosporin. Therefore, when we talk about the members of this gram positive bacteria, you have Streptomyces, Actinomyces. All those are the examples. Just the opposite is gram negative bacteria. They do not retain the gram stain. Moreover, the cell wall consists about around 18% peptidoglycan, but outer membrane is present and lipopolysaccharides are present. They have these characteristics that they are resistant to antibiotics. So when we suffer from any kind of diseases, if it is a gram negative bacteria, it takes a longer time to overcome. Clear? So that was all about gram positive and gram negative bacteria. As you have studied in the previous classes in class 9 and all, you must be knowing that bacteria can be grouped according on the basis of their shape into 4 types. Let me show you. Those are that is a rod shaped bacteria. First let me write rod shaped bacteria, which are referred as bacillus. Then you have coccus, a round shape. Then you have vibrio, that is comma shape. And then you have spiralium or spiral shape. These are the 4 types of shapes of bacteria. Clear? So, when we talk about the rod-shaped bacteria, which is also referred as bacillus, here you find that these are rod-shaped. They may possess flagella or may not possess flagella. Then you have the coccus, which are round shape. If there are two, diplococcus. If they are present in a chain, streptococcus. If they are present in group, it is referred as staphylococcus. You have got the example diplococcus pneumoniae. First one is lactobacillus, one example. Vibrio chlori, that is an example of comma-shaped bacteria, and spirillum bacteria, which is spiral shape. Clear? Now, again we come to the diagram. See, here you have one more features, that is like we were discussing about the cell envelope. Cell envelope consists of glycocalyx or mucilage sheath or capsule. Then we have the cell wall and the innermost plasma membrane. So the plasma membrane, as you know, allows the semi-permeable, allows the entry and exit of substances and maintains the homeostasis. Clear? Now there are other uh, structures also that you can see apart from the cytoplasms. There are enfoldings which is called as mesosomes. Clear? Since membrane bound organelles are absent in the prokaryotic cell, these mesosomes take over the function of aerobic or anaerobic respiration. Clear? They are involved in respiration process since mitochondria is absent. Apart from it, in certain bacterial cell like cyanobacteria or blue-green algae, photosynthetic pigments called chromatophores are present. Clear? Then you have one most important features called as the flagella, also known as singular flagellum. Now, this flagella has got three parts: basal portion, hook, and the elongated portion called as flagellum. Now, the basal portion is a round dot-like structure which may be two ring in gram-positive, that is two swellings, or if it is a gram-negative bacterial cell, the basal body has got four rings or swellings. Then just above it, you have a hook-like structure. Got it? That is made up of protein subunits. And the uppermost part, elongated tubular structure called as flagella, made up of protein flagellin. This you may be asked what describe the different parts of flagella. Apart from it, you have got certain inclusions which are called as gas vacuoles, which are permeable to gases but not to water. Then you have got glycogen granules. Then we have only one organelle apart from nucleus that is important, found in other eukaryotic cell, that is ribosomes. Now what are ribosomes? These are structures which are involved in protein synthesis. Now when we talk about ribosomes, in bacteria it is of 70S type. Clear? It has got two subunits, one smaller subunits and one larger subunits. And you have got many ribosomes attached to a strand of mRNA, that is called as polyribosomes. So sometimes this is also asked, what do you mean by polyribosome? Poly means many. So when there are about more than 4 to 8 ribosomes attached to a strand of mRNA, that is messenger RNA, it is called as polyribosome. What is the function? It translates the information into protein. Clear? Then that is, anything else is left? I don't think so. So ribosomes, yes, one more important features apart from this, since this, as I said, that sometimes there is an extra chromosomal structure called as plasmid. You have structures called as fimbri or pili. Pili are structures which are made up of protein pilin, and they act as conjugation tube. Bacteria undergoes reproduction by three methods, that is binary fission, transduction and conjugation, sexual conjugation. So in that case what happens, two bacterial cell, they come together. Clear, this is one bacterial cell, and this, they form a conjugation bridge. So this conjugation tube is formed by a structure called as pili. Clear? Apart from that, you have got one more structure called as fimbriae, not found in all the bacterial cell. These are solid structures, have bristle-like outgrowths that helps the bacterial cell to attach to any solid substratum. Since bacteria are found in almost almost all type of habitat except fire. So let me give you the notes, this diagram. Then cell, the unit of life. So these all comes contributions, you should know. These are some of the important ones. Then which is the smallest bacterial cell, which is the largest, what do you mean by omnious cellular, who proposed it? This is the cell envelope, which consists of three parts: glycocalyx or mucilaginous sheath or capsule. Then you have the cell wall and plasma membrane. Christian Gann was the person who differentiated the gram-positive and gram-negative bacteria on the basis of the staining. Crystal violet dye, iodine, and then treated with alcohol. Other features include the nucleoid, mesosomes, chromatophores, flagella.